Welcome my dear students. Today we are entering one of the most fundamental yet truly fascinating chapters of biochemistry, that is the chemistry of water. Though it looks simple, this tiny molecule is the reason life exists, functions and evolve. In this class on biochemistry of water, properties, interactions, ionization and buffers, we will discuss how water molecules behave, how they interact with other biomolecules, why weak bonds matter, and how water maintains pH balance through buffering systems. Just like a master conductor keeping every part of an orchestra synchronized, water controls structure, stability, and reactions inside living cells. Whether you are a PG or UG student or preparing for CSIR-NET Life Science or GATE, understanding these concepts will give you a strong foundation for advanced biochemistry. So settle in. We are about to explore how this seemingly simple liquid supports complex life with remarkable precision. So the first is the polarity of water. The structure of molecule, water has a bent molecule as the image is showing here, with a bent molecular shape with the oxygen atom at the center and two hydrogen atoms at an angle of approximately 104.5 degree. Charge distribution, the oxygen atom is more electronegative, pulling shared electrons. We know the equal sharing of electrons is found in case of covalent bond formation. So this is the hydrogen oxygen covalent bond. So the shared electrons are pulled towards the oxygen, closer to the oxygen, creating a partial negative charge on oxygen and partial positive charge on the hydrogen atoms. Biological significance, the polarity allow water molecules to attract and interact with other polar molecules and ions, enabling chemical reactions essential for life. Hydrogen bonding, nature of hydrogen bonds, each water molecule can form up to 4 hydrogen bonds, 2 through its hydrogen atom and 2 through its oxygen atom. We know that. Effects, cohesion, adhesion. Water molecules stick to each other, resulting in a phenomena like surface tension, that is called as cohesion. And when water molecule binds to other substance, aiding processes like capillary action in plants, it is called as adhesion. So biological significance, both the cohesion and adhesion enable water transport in plants and the maintenance of a stable water column in xylem vessels. High specific heat. Water can absorb large amounts of heat with minimal temperature change. This is due to the energy required to break hydrogen bonds before molecules can move freely. Effects, thermal stability. Water stabilizes temperature in aquatic ecosystems, providing a stable environment for organisms. It helps maintain body temperature in organisms as water resists sudden temperature fluctuations. High heat of vaporization. We know that water requires significant energy to transition from a liquid to a vapor state, as hydrogen bonds must be broken. So the effect is cooling mechanism. Evaporation of water, that is sweating or transpiration in plants, absorb heat from the body or surrounding, aiding in temperature regulation. Climate regulation. Large bodies of water moderate climate by releasing heat slowly during the cooling phase. Density anomaly, we know when water freezes, its molecules arrange into a crystalline lattice due to hydrogen bonding. This structure spaces molecules further apart than in liquid water. Effects, lower density, ice is less dense than liquid water, allowing it to float. Insulation, floating ice forms a protective layer on water bodies, insulating the liquid below and enabling aquatic organisms to survive in cold climates. Solvent properties, universal solvent. Water's polarity allows it to interact with and dissolve ionic compounds like NaCl and polar molecules like sugar. Hydration cell, What water molecules does? Water surrounds individual ions or molecules, separating them and preventing them from further reassociation. So the biological significance of the solvent property is the enabling the transport of nutrients, gases and wastes in organism, and it serves as a medium for biochemical reactions essential for metabolic processes. Additional properties of water, that is the first of all is the transparency. Water is transparent and it allowing sunlight to penetrate aquatic ecosystem and enabling photosynthesis in underwater plants. High surface tension due to cohesion of molecules, water can resist external forces, enabling insects like water striders to walk on its surface. Hydrogen bonding. We know hydrogen bonds is a dipole-dipole interactions and that occur when a hydrogen atom covalently bonded to an electronegative atom such as an oxygen or nitrogen. So hydrogen bonds is a weak bond found in water molecules and it interact with another electronegative atom. Role in biomolecules. It stabilizes secondary structures of proteins such as alpha helicase and beta sheet by forming bonds between carbonyl oxygen and amide hydrogen. It maintains the tertiary structure of proteins by forming hydrogen bonds between side chains of amino acids. The nucleic acid hydrogen bonds between complementary base pair stabilize the double helical structure of DNA. It contributes to the recognition and specificity of interactions in enzyme substrate binding and antigen antibody interactions. Importance in water. Hydrogen bonding gives water its high boiling point, cohesive properties and ability to act as a solvent for polar and charged molecules. Ionic interactions. Electrostatic forces between oppositely charged ions or between charged groups on molecules. Characteristics, they are stronger than hydrogen bonds but weaker than covalent bonds. Depending on the dielectric constant of the surrounding medium, which is higher in water, reducing the strength of ionic interactions. Role in biomolecules, it stabilizes the tertiary and quaternary structures of proteins through interactions between positively charged residues and negatively charged residues. It facilitates binding of proteins to DNA via interactions between negatively charged phosphate group in DNA and positively charged amino acid residues in the protein. So it is important in ion channel where ionic interactions help regulate ion flow across the membrane. In solution, it is dissolved ions like Na plus and Cl minus interact with water molecules, affecting osmotic balance and biochemical reactions. This is the ionic bonding. Van der Waals forces, weak non-specific forces that arise from temporary dipoles created by fluctuating electron distributions in molecules. Type, there are basically two types of van der Waals forces, London dispersion forces and dipole-dipole interactions. It results from transient dipole in non-polar molecules. Dipole-dipole interactions occur between polar molecules with permanent dipole. Role in biomolecules, it stabilizes close packed regions in proteins and other macromolecules. It is essential in ligand binding to receptors by facilitating complementary surface interactions. Significance. These forces act over very short distances but it is additive, significantly contributing to the stability of macromolecular assemblies like protein-protein interfaces. Hydrophobic interaction, that is another kind of weak interactions found in water. Nonpolar molecules or group clustered together in aqueous solution to minimize disruption of hydrogen bonded water network. Mechanism, water molecules form ordered cages or cell around nonpolar molecules which is energetically unfavorable, and aggregation of nonpolar molecules reduces the surface area exposed to water, increasing entropy and stabilizing the system. Role in biomolecules, it drive protein folding by positioning nonpolar side chains in the interior of the protein and polar side chains on the exterior. This is critical for the formation of biological membranes where hydrophobic tails of phospholipid aggregate to form bilayers, leaving hydrophilic heads exposed to water molecules. It also stabilizes lipid-protein interactions in membrane and support the self-assembly of micelles and vesicles. Ionization of water. The ionization of water is a fundamental chemical process in which water molecules self-ionize to produce hydronium ion and hydroxide ion. This dynamic equilibrium plays a critical role in maintaining the acid-base balance in biological and chemical systems, such as the self-ionization process equation involves that the 2H2O, it reveals that H3O plus OH minus, and alternatively it can be simplified as H2O, that is H plus OH minus. So the mechanism: a small fraction of water molecule, approximately 1 in 10 to the 7 molecules at 25 degrees Celsius, undergoes ionization. So one water molecule donates a proton to another, forming a hydronium ion, that is H3O plus, and a hydroxide ion, that is OH minus. This is a reversible process and establishes a dynamic equilibrium. Ion product of water, or Kw. The ion product of water, that is Kw, is the equilibrium constant for the self-ionization of water. It is given by Kw is equal to H plus into OH minus. Value at 25 degrees Celsius. At standard temperature or 25 degrees Celsius, Kw is equal to 1 into 10 to the minus 12 mole square per liter square. This value is temperature dependent and increases with rising temperature. Neutral water. In pure water, the concentrations of H plus and OH minus are equal. That is, H plus is equal to H minus is equal to 1 into 10 to the minus 7 mole per liter. So the ion product of water, or Kw, at 25 degrees Celsius is the H plus into OH minus is equal to 1 into 10 to the minus 14. pH. pH is basically a measure of the hydrogen ion concentration, that is pH is minus logarithm of H plus. Neutral water has a pH of 7. Importance: the pH of a solution affects enzyme activity, ion solubility and biochemical reaction rates. Buffer. Key to pH stability. Buffers are crucial or critical system in chemistry and biology that resist changes in pH upon the addition of small amounts of acids or bases. They play a vital role in maintaining pH homeostasis in biological and chemical environments. How buffers work? A buffer is a solution that minimizes pH fluctuations when an acid or base is added. Mechanism: buffers typically consist of a weak acid and its conjugate base, or a weak base and its conjugate acid. So the weak acid neutralizes added bases by donating a proton, while the conjugate base neutralizes added acids by accepting a proton. So the example: the acetic acid CH3COOH and acetate ion CH3COO minus system. This is the buffering system. Buffer components: a weak acid and its conjugate base, that is acetic acid and acetate ion. Buffering capacity: the buffering capacity is the ability of a buffer to resist the pH changes depending on its concentration and proximity of the solution's pH to the acid's pKa. Maximum buffering capacity occurs at the weak acid's pKa value, where acid is equal to base. Buffer effectiveness diminishes significantly when pH deviates more than 1 unit from the pKa. Factors affecting buffering capacity: concentration. Higher concentration of the acid and conjugate base increase the capacity. Ratio of components: we know that a balanced ratio of acid to base provides better buffering efficiency. Biological buffers. Buffers are very much crucial for maintaining the soluble pH required for enzymatic activity, metabolic processes, and structural integrity of various biomolecules. Some key biological buffer system include bicarbonate buffer system, primarily operates in the blood plasma. It maintains blood pH. Impairment of this system leads to acidosis or alkalosis, conditions that disrupt normal physiological function. Phosphate buffer system. The phosphate buffer system is one of the critical buffering systems in living organisms, particularly effective in intracellular fluids. It maintains the pH balance within cells by resisting drastic changes in hydrogen ion concentration. Its function in intracellular fluids: it plays a key role in maintaining the pH of the cytoplasm and other intracellular compartments, ensuring enzyme functionality and metabolic efficiency. In the kidneys, the phosphate buffer system helps maintain blood pH by excreting H plus ions as dihydrogen phosphate in urine. The system is also integral to the phosphate group in energy-rich compounds like ATP, where the interplay between the different phosphate species is central to energy metabolism. Protein buffers. Proteins act as an effective buffer in biological system due to the presence of amino acid side chains capable of accepting or donating hydrogen ion. The buffering capacity actually provides stabilized pH in intracellular and extracellular fluid, playing a vital role in maintaining homeostasis. And proteins like hemoglobin in red blood cells play a critical role in buffering blood pH. Hemoglobin binds H plus ions during oxygen delivery, maintaining pH balance. Cytoplasmic proteins stabilize intracellular pH, ensuring optimal enzyme activity. Protein buffers complement other buffering systems like bicarbonate and phosphate, providing a broad range of buffering across different pH levels. Buffers so ensure the proper folding and function of proteins, or as drastic pH changes can lead to denaturation. Examples of protein buffer: hemoglobin first of all act as a buffer in blood by binding H plus ions to the imidazole group of histidine residues. This mechanism is essential in the Bohr effect. Hemoglobin releases more oxygen readily at lower pH. Albumin, the most abundant blood plasma protein with multiple ionizable side chains, contributing to the blood buffering. Enzyme and structural proteins. We know intracellular proteins maintain localized pH for cellular reactions and structural integrity. So my dear students, we have now uncovered how water, the most abundant molecule in living organisms, shapes biology at every level. From its extraordinary properties and weak interactions to its ability to ionize and stabilize pH through buffers, water truly acts as the chemical framework that makes life possible. As you prepare for university exam or national level tests like CSIR-NET or GATE, set, remember that these basic principles are the backbone of many higher biochemical processes. Thank you so much for joining this class. Keep revising, stay curious, and continuous exploring beautiful chemistry that keeps life running smoothly. See you in the next class. Bye.